you can tell, but we share Bard's pain of having a small team. But so far, Craig and Michelle have told you about the different software tools that our computational team came up with, which includes Genome Surfer Pro, Optimus Primer, Trumpet, the Puppet Show, the eLab Notebook, and the last section is the other subset of our team, which is the wet lab section. We weren't so much interested in creating these software tools, but using them to solve biological problems. The biological problem that we chose to look at was tuberculosis. Unless you've been hiding under a rock, you know tuberculosis is bad. You can also see in the map that it's a global problem. So besides being big and bad, it is also very complex. So you can see in the gene regulatory network here that there's many different interactions between the genes. This is a perfect example of what Craig was talking about before, where we can use the invertases to study how they interact with each other using one plasmid. So here's just a refresher again that shows you, if the gene's turned off, you can add the invertase and it'll turn it back on so you can study the different interactions. So the next question would be, well, how would we go about doing this with all the tools you just mentioned? Well, the first thing you'd have to do is you'd have to pick the genes you want, and we would do that using Genome Surfer Pro. The next thing you would do is to create the primers. We can do that with Optimus Primer. After we do that, we need to create a roadmap that will tell us, well, where do we put the invertases in and all those different combinations, and we have Trumpet for that. The next thing we do is we'd actually create them. We had two options available to us in the lab this summer. We were able to construct it manually using regular protocols, or we had Puppet Show in conjunction with the liquid handling robot TCAN. After we create them, we need some place where we can store all the parts that we use to make these constructs, as well as the actual constructs themselves. Clotho has a suite of tools that allows us to do so. This summer, we first started off by creating a bunch of fluorescent protein devices. We used a variety of promoters, including inducible, constitutive, and repressible. Like I already mentioned, we used both the manual protocol, which you can see here, and we did that manually. And the other way we did it was using the TCAN robot shown here, and we used the Puppet Show code, which is here, that corresponds to the protocols that we were using by hand. Down here, you can see an example of a gel showing restriction digest. Lane 1 is an uncut plasmid, whereas column 2 shows you the results of doing the restriction digest by hand. As you can see through columns 3 through 5, which were done using the robot, there's no difference. Lastly, we used the fax machine to characterize our data. We also went out and got involved in the community. We went to Somerville High and we talked a little bit about synthetic biology. We came up with a board game that closely mimics Scrabble in order to continue teaching. You can think of it as the genes as the letters, and the genetic device you create is the word, and you get points depending on the word you created. And Wellesley reached out to their community and put on a poster for their a poster presentation.
So in terms of future work, the most exciting thing for the HCI lab and the front-end programmers is to fully implement the Trumpet and electronic lab notebook that we started working on this summer. We can't wait to test those out in the field.
As far as Trumpet is concerned, in the future, we know that invertases don't work 100% of the time. There's some probability that they will make this flip and some that they won't. We hope that people will start to characterize invertases and will be able to incorporate this information into Trumpet so that the user can choose the right invertases to use. And so we'd just like to quickly summarize what exactly we've done this summer. Michelle at the beginning of the presentation said that we wanted to make this design flow completely automated using a suite of tools. So we have a collaborative tabletop research tool. This is our Genome Surfer Pro. This allows you to study your genes and choose the right one, and then you can use Optimus Primer to turn these into our biobricks. We have created 2 different algorithms which allow us to place invertase sites so we can make reconfigurable genetic circuits using Trumpet. We have also created Puppet Show, which allows us to automate lab protocols. And not only have we made all this software, but we've been able to successfully test this software in the wet lab, and we made sure that we're making these tools for the wet lab members, and it's not just a bunch of computer science tools that nobody's going to use. We'd of course like to acknowledge all of our sponsors. We couldn't have done this without their help. And likewise, we really appreciated all the help that we got from our mentors. They've been a great source of inspiration for us during this project. And now we'd like to open it up to questions. Thank you.